Hi! Good day, everyone. Welcome to Microbiology. This is our session 3. Ever since the 1977 proposal by Carl Woese and George Fox to group life into 3 domains, bacteria, archaea, and eukarya, new discoveries with the archaea keep emphasizing their unique biology. Very novel lipids, lack of peptidoglycan, and many eukaryotic-like traits are just some of the fundamental properties that set archaea apart from the other domains of life. However, recent studies have shown that even some housekeeping biochemistry in archaea differs from that in the other domains, and a good example is the metabolism of sugars. Studies of sugar metabolism in archaea have focused on species in the phylum Crenarchaeota, most of which grow at extremely high temperatures, and the phylum Euryarchaeota, many of which, like the cells of Halocucus as shown in the picture, grow at extremely high salt concentrations. Several species in these two phyla can use glucose and some other sugars as carbon and energy sources. But whether sugars are used or not, all archaea must be able to metabolize sugars because they form the backbone of cell structures such as cell walls and macromolecules such as nucleic acids. Microbial nutrients and nutrient uptake. Metabolism is the series of biochemical reactions by which the cell breaks down or biosynthesizes various metabolites. In order to grow, cells must incorporate nutrients from their environment, transform them into precursor molecules, and then use them to construct a new cell. In this section, we will examine some of these processes, with a focus on these areas. 1. Defining the basic nutrients of life. 2. Exploring seminal metabolic pathways and alternative metabolic lifestyles. 3. Biosynthesizing the building blocks of macromolecules. Feeding the microbe. Cell nutrition. Because the metabolic capacities of microbes differ, their nutrient requirements also differ. However, all microbes require a core set of nutrients. Some nutrients, called macronutrients, are required in large amounts, while others, called micronutrients, are required in minute amounts. Just a handful of the chemical elements predominate in living systems: hydrogen, oxygen, carbon, nitrogen, phosphorus, sulfur, and selenium. Carbon is needed in the largest amount, comprising 50% of a cell's dry weight. Oxygen and hydrogen are next, when combined, comprising 25% of cell's dry weight, and nitrogen follows, which is 13%. All the required phosphorus, sulfur, potassium, magnesium, and selenium combine for less than 5% of a cell's dry weight. In addition to this, at least 50 other elements either are required by one or more microorganisms or, if not required, are still metabolized in some ways. Elements in row 7 are not known to be metabolized by microorganisms, and elements beyond that. Microorganisms require several metals in very small amounts relative to macronutrients. Chief among these is iron, which plays a major role in cellular respiration. Besides iron, many other metals may be required or otherwise metabolized by microorganisms. Collectively, these metals are called trace metals and typically function in the cell as cofactors of certain enzymes. Table 3.1 lists the major trace metals and other trace elements of life and examples of enzymes or other molecules in which each is found. Growth factors differ from trace metals in that they are organic rather than metallic micronutrients. Common growth factors include the vitamins, but amino acids, purines, pyrimidines, and several other organic molecules may be growth factors for one or another microorganism. Vitamins are the most frequently required growth factors, and a few common ones are shown in the table. Most vitamins function as enzyme cofactors. Vitamin requirements vary among microorganisms, ranging from none to several. Lactic acid bacteria of the genera Streptococcus, Lactobacillus, and Leuconostoc inhabit nutrient-rich habitats, such as dairy products and the animal gut. These bacteria are well known for their vitamin requirements, which are even more extensive than those of humans. If a cell is to grow and divide, it must take up its macronutrients and micronutrients from the environment. But this process is not as easy as it may seem, primarily because of the impermeability of the cytoplasmic membrane and the fact that the concentration of a given nutrient in the cytoplasm must often be much higher than its concentration in the environment. How does nutrients are transported to the bacterial cell? In this section, we will discuss about the active transport and transporters, simple transporters and group translocation, periplasmic binding proteins, and the ABC system. The three classes of transport systems. Note how simple transport and ABC system transport substances without chemically modifying them, whereas group translocation results in chemical modification, in this case, phosphorylation of the transported substance. The three proteins of the ABC system are labeled 1, 2, and 3. Simple transport consists only of a transmembrane transport protein. Group translocation employs a series of proteins in the transport event. And the ABC transport system consists of three components: a substrate binding protein, the transport membrane protein or the transmembrane transporter, and an ATP hydrolyzing protein. Each of these transport systems is energy-driven, be it from the proton motive force, ATP, or some other energy-rich compound. Simple transport reactions are driven by the energy inherent in the proton motive force. The major transport events catalyzed are either symport reactions, where a solute and a proton are co-transported in one direction, or antiport reactions, where a solute and a proton are transported in opposite direction. One classic example of a simple transporter is the uptake of a disaccharide sugar lactose by way of the lac permease, which is a symporter. This symporter is a well-studied symporter in Escherichia coli. As each lactose molecule enters the cell, the potential energy in the proton motive force is diminished slightly by the cotransport of a proton. The net result is the energy-driven accumulation of lactose in the cytoplasm against the concentration gradient. Many other solutes enter by the activity of their own simple symporters, including phosphate, sulfate, and several different organic compounds. Mechanism of the phosphotransferase system of Escherichia coli is shown in the picture. For glucose transport, the system consists of 5 proteins. We have enzyme 1, enzyme 2A, 2B, 2C, and the HPR. A phosphate cascade occurs from the phosphoenol pyruvate to enzyme 2C, and the latter actually transports and phosphorylates the sugar. Group translocation differs from simple transport in two important ways. 1. The transported substance is chemically modified during the transport process, and 2. An energy-rich organic compound rather than the proton motive force drives the transport event. The best-studied group translocation systems transport the sugars glucose, mannose, and fructose in E. coli. During uptake, these compounds are phosphorylated by the phosphotransferase system. The phosphotransferase system consists of a family of 5 proteins that work in concert to transport any given sugar. Before the sugar is transported, the proteins in the phosphotransferase system are themselves alternately phosphorylated and dephosphorylated in a cascading fashion until enzyme 2C phosphorylates the sugar as it enters the cytoplasm. A protein called HPR, the enzyme that phosphorylates HPR enzyme 1 and enzyme 2A, are all cytoplasmic proteins. By contrast, enzyme 2B is a peripheral membrane protein and enzyme 2C is the transmembrane component. In the phosphotransferase system, HPR and enzyme 1 are non-specific components and participate in the uptake of several different sugars. By contrast, distinct enzyme 2 proteins exist, one set for each different sugar transported. Energy to drive the phosphotransferase system comes from phosphoenol pyruvate, an energy-rich intermediate in glycolysis. The image shows the mechanism of an ABC transporter. The periplasmic binding protein has high affinity for substrate. The membrane-spanning protein form the transport channel, and the cytoplasmic ATP hydrolyzing protein supply the energy for the transport event. Gram-negative bacteria contain a region called the periplasm that lies between the cytoplasmic membrane and the outer membrane. The periplasm is home to proteins that carry out different functions, including transport. The latter is catalyzed by the activity of periplasmic binding proteins. Transport systems that employ a periplasmic binding protein along with transmembrane and ATP hydrolyzing components are called ABC transport systems. The ABC standing for ATP binding cassette, a structural feature of proteins that bind ATP. More than 200 different ABC transport systems have been identified in various bacteria, and these catalyze the uptake of a wide variety of organic and inorganic compounds. Although gram-positive bacteria and most archaea lack a periplasm, they also have ABC transport systems. In gram-positive bacteria, substrate-binding proteins are attached to the external surface of the cytoplasmic membrane. Once they bind their substrate, these proteins interact with their transmembrane component to catalyze the ATP-driven uptake of the substrate. ABC systems are also present in a variety of archaea and are primarily employed for the transport of sugar. Since you already have the idea on the transport of nutrition in bacterial cells, let us move on to the energy classes of microorganisms. The picture shows the metabolic options for conserving energy by microorganisms. Energy sources can either be chemical or in light form. Most organisms employ only one option, but some have two, and a few rare species can tap into all three forms of energy conservation. Energy classes of microorganisms. These are grouped into chemoorganotrophs, chemolithotrophs, and phototrophs, heterotrophs, and autotrophs. Organisms that conserve energy from chemicals are called chemotrophs, and those that use organic chemicals are called chemoorganotrophs. Most organisms in laboratory culture are chemoorganotrophs. A wide variety of organic compounds can be catabolized during chemoorganotrophic metabolism, and some of the energy released during their oxidation is conserved in the energy-rich bonds of adenosine triphosphates or ATP, the cell's energy currency, or related energy-rich compounds. The picture shows the crystal violet stain of Escherichia coli and an E. coli observed using a transmission electron micrograph. Many bacteria and archaea can tap the energy available from the oxidation of inorganic compounds, and this form of catabolism is called chemolithotrophy. Organisms that carry out chemolithotrophic reactions are called chemolithotrophs. Several inorganic compounds can be oxidized. For example, gaseous hydrogen, hydrogen sulfide, ammonia, and ferrous iron. Related groups of chemolithotrophs typically specialize in the oxidation of a group of related inorganic compounds, and thus we have the sulfur bacteria, the iron bacteria, and the nitrifying bacteria, and so on. One example of a chemolithotroph is the Theobacillus thiooxidans. Phototrophs contain chlorophylls and other pigments that convert light energy into ATP, and thus, unlike chemotrophs, do not require chemicals as a source of energy. Two forms of phototrophy are known in bacteria. In one form, called oxygenic photosynthesis, oxygen is produced. Oxygenic photosynthesis is characteristic of cyanobacteria, a major lineage of bacteria, and is also carried out by algae. The other form of phototrophy, anoxygenic photosynthesis, occurs in at least 6 phylogenetic lineages of bacteria, including the purple and green bacteria, the heliobacteria, and many others. In anoxygenic photosynthesis, oxygen is not produced. This image is a picture of Rhodobacter capsulatus, which is an example of phototroph. Regardless of how a microorganism conserves energy, be it from chemicals or from light, all cells require large amounts of carbon in one form or another to make new cell materials. If an organism is a heterotroph, its cell carbon is obtained from one or another organic compound. An autotroph, by contrast, uses carbon dioxide as its carbon source. Chemoorganotrophs are by definition also heterotrophs. By contrast, most chemolithotrophs and phototrophs are autotrophs. Autotrophs are also called primary producers because they synthesize new organic matter from inorganic carbon. Virtually all organic matter on Earth has been synthesized by primary producers, in particular the phototrophs. The Calvin cycle is the major biochemical pathway by which phototrophic organisms incorporate carbon dioxide into cell material. That is all for this session. Stay safe and God bless to all of you. See you in our next one. Bye!